So why don't we start out reviewing the last of the notes from chapter 10.2 or the PowerPoint 10.2, stuff I did in the last class, and then we'll segue into the weekend quiz, and we can talk about the problems I posted. If we have any time left, I'll start the next section of notes. So let's do that. Share my screen. Okay. Cancer biology. Okay, I think where we picked up last time was right about here, if I'm not wrong. Well, let's back up to here. Viruses. Okay. Viral causes of cancer. So, as I said, there aren't a whole lot of cancers that are caused by viruses. Two really important ones here. One is HPV, and you can see its life cycle there. What I find interesting about HPV is the way it evolved two proteins that do just what you need to do to get the cell to keep dividing. Blocking RB, which releases E2F, and blocking P53, which stops the apoptosis that would happen if it just released E2F. Epstein-Barr takes a different path here. It has two receptors associated with it. I don't know. I didn't look into the background, but I wouldn't be surprised if these were homologs of some human proteins that were picked up by the virus when it jumped into and out of the genome. They tend to do that. They'll jump into the genome and then grab the gene that's next to it when they jump out, and then that gene becomes part of their genome. It's probably where these came from, because they operate the cell just the way you would hope a normal receptor would. It's going to activate BCL2 over here to inhibit apoptosis, activate cell division through NCAFA B, which we didn't talk about, and other pathways. It also has this one that's activating RAS, RAF, and through that whole pathway. Question?
I was just wondering, why wouldn't the retroviruses cause cancer if they're mutating our DNA? In terms of is it because it has more whole body physiological effect versus this affecting specific tissue?
Yeah, I think they can cause cancer. The thing is, every time they insert into a genome, they're going to insert in a different place. It's not specific. So say you've got, I'm just going to make up numbers here. Say you've got 10,000 viruses infecting 10,000 cells. One of them ends up cancerous. The immune system takes it out. When it becomes dangerous is when all 10,000 viruses are activating cancer in 10,000 cells, right? And then it's going to, possibly get around the defenses of the cell.
So if the retroviruses were to infect the proto-oncogene or the oncogene, then it can lead to cancer, whereas if it's other part, it doesn't cause cancer.
Yeah, right. Yeah, you got to remember, most of the genome is, we used to call it nonsense DNA, but non-coding DNA anyway. And so if it ends up in one of those regions, then it's not going to cause any problems in the short term. Okay. So proto-oncogenes. Basically, what is a proto-oncogene? It's a gene for any pro-growth regulator in the cell. And once it becomes mutated, so it's no longer in control, it's an oncogene. So any pro-growth regulators, so we're talking growth factors, receptors, G proteins, kinases, transcription factors, and cell cycle regulators. So everything from outside the cell to your DEGs, the delayed early genes, can be proto-oncogenes and can become oncogenic. And then we talked about how proto-oncogenes become oncogenic. One way is by point mutation. So point mutation causing a constitutively active protein. And again, remember, most mutations are going to be either going to knock out the function of the gene or have no activity whatsoever. But there are certain amino acids that you can knock out that will constitutively activate certain proteins. So RAS in particular is a great target for this. We've got 1, 2, 3 different amino acids that can be changed. And when they're changed, RAS no longer interacts with GAP, and so it never shuts off. Here's a mutation that took out the ligand binding region of a protein, of a receptor tyrosine kinase, and that causes it to become constitutively active. Overexpression, another popular way to make a proto-oncogene into an oncogene. You can get gene duplication, which you can get multiple copies of a gene. And when you have multiple copies, you end up with more of the protein. And things like receptor tyrosine kinases, when they're overexpressed, they're going to just start to activate each other because there's so many of them, they bump into each other. And it's the bumping together that causes them to activate. So that can happen with overexpression. And, you know, pretty much any other protein that's a pro-growth regulator, if overexpressed, is going to cause problems. Translocation, when we get a chunk of one chromosome switching with the chunk of another chromosome. Like this, here's chromosome A gets cut, chromosome B gets cut, they swap over. This is non-homologous recombination. It can happen during mitosis, during S phase, I mean, not during mitosis. It can happen during S phase, whenever you're replicating. Here's an example how that could be bad. Burkitt's lymphoma. Your MYC gene, which normally in these white blood cells would be quiet because they're post-mitotic. So they, you know, they're not stem cells anymore. They've gone down that pathway. They should be post-mitotic. So this part of the DNA is all condensed up and not active, as opposed to this region, white blood cells making amino antibodies are going to need heavy chains. So this is a really active region. Switch those two chunks, and now you've got MYC in an active region. It gets overexpressed, and those cells then become cancerous. Here's another kind of interesting translocation where you've got a big chunk of 9 switching with 22, and as a result, you get a whole new chromosome here, which we call the Philadelphia chromosome, and that makes a new protein, BCR-ABL. So BCR is this section, and then this gets hooked onto ABL, and you have this new protein, which is a kinase that is no longer regulated, BCR-ABL, and that causes chronic myelogenous leukemia, which can go to acute myelogenous leukemia. Local DNA arrangement. So we're talking within the same chromosome, a big chunk gets cut out or a big chunk inverts. And this is another neat one in that one example of this inversion is this tropomyosin nerve growth factor receptor, which when it flips around, you end up with the nerve growth factor with tropomyosin on it. And so it's constitutively active because it binds to itself that way. And so this is where we're getting with Tillich's question there. Viral DNA can insert next to a proto-oncogene, and that turns it into an oncogene, so that can happen as well. And then we get into our tumor suppressors, 2 different types of tumor suppressors, gatekeepers and caretakers. Caretakers are the ones that are fixing DNA, sensing damage and fixing the DNA, and your gatekeepers are the ones that are blocking cell cycle directly. So APC, we talked about quite a bit, part of the destruction complex. P10, which is converting PIP3 into PIP2. RB, SMAD4, part of TGF-beta signaling, which is usually inhibiting cell division. P53, and these things here. And there's an example of a gatekeeper gone, right? Not gatekeeper, I'm sorry, caretaker knocked out, xeroderma pigmentosa. Cause of mutation, cancer. See with my undergrads, I usually promote smoking with them because they're not going to, if they live longer, their Social Security is going to be gone anyway. So just enjoy your life. You're only going to enjoy 65 years. So Just saying, just saying. Okay, cancer bad. So a lot of carcinogens in cigarettes, but also the liver can convert some things into carcinogens. So they would be considered pre-carcinogens until your liver converts them. Ionizing radiation, another source of mutations. Ultraviolet is non-ionizing, but it causes thymine dimers. And then X-ray and gamma ray are much more dangerous, which is why space travel is dangerous, right? We're not being, the astronauts are not protected from the background gamma radiation the way our atmosphere does. Thank you, atmosphere. And then there's two different kinds of carcinogens. You've got your initiators and promoters. Initiators activate, cause the DNA damage that turns certain genes on. And then the promoter then, together with the initiator, can cause cancer to grow. Stepwise development of cancer. So typically, one mutation, two mutations in a cell isn't going to make it an incredibly aggressive cancer. But if the cancer is allowed to grow and grow and grow, it will, because it's lost a lot of caretaker genes, you get more and more DNA mutations, and you get more and more aggressive cancers. Where am I going with this? Stepwise development of cancer again. So I started out talking about how the colon cells are being replaced normally, and it has to do with Wnt signaling. So if we knock out APC, cells start to grow more, and then you pick up other mutations like RAS, and then P53 and VAX, and they get more and more aggressive. Question?
Usually when there's a cell development, it usually goes out with the dead cells, basically, like how it does with our skin and all of that. How does it causes it to stick within one another in terms of colon cancer?
Like to become a tumor, right?
Yeah. Versus the cell dying and leaving off.
Oh, sloughing off
Yeah.
Instead of, because they don't mature. They stay in this dedifferentiated state that we talked about earlier. So as the cells move up this crypt, they mature, and then they become adult, and then part of that process is them senescing and dying and then being replaced. So these are dedifferentiated. So they're like stem cells. So they're just going to keep dividing. This is just talking about APC, what happens without it. And then our hallmarks of cancer. Self-sufficient growth signals, and that's really the key to everything, right? Because that's going to turn on all the other things. Insensitive anti-growth signals like TGF-beta, SMAD4 mutation, evading apoptosis, limited replicative potential, which is our telomerase, sustained angiogenesis, bringing in blood, and tissue invasion and metastasis. And that's when it is very dangerous when it does that. So that was the review of what we talked about last time. Let me see if I can bring up our quiz. Test correction, don't forget exam 3. You can go ahead and do that. We'll probably do it for exam 4 too. What the hell? That way you're learning more. You're not just moving on. Feeling a little uncomfortable, frankly, about having 10 multiple choice questions, because you miss one, that's 5 points, right? That's a lot. Maybe I'll put up a quiz, not a quiz, a poll, and see if you want more questions. It always seems like if I say this, then other people don't want that, but whatever. We'll see what we can do. Okay, I'm babbling now. I'm sorry. Let us do this quiz. Once again, Dr. Slish is on the point here. See if I can get my own quiz right. Which of the following is not a hallmark of cancer? anchorage dependent growth, loss of density, angiogenesis, dedifferentiation. So basically, loss of anchorage dependent growth would be cancer. So that is the answer there. What's your role of P10? It's going to dephosphorylated PIP3, and that's a caretaker.
I think you kind of went over this when we were talking about the hallmarks, but can you just define what you mean by anchorage dependent growth?
Sure. Let me, as long as I have that PowerPoint. Okay, so anchorage dependent growth means that the integrins have to be attached to extracellular matrix in order to stay alive. And when they lose that, they'll typically go into apoptosis. So they have to get over that and get into anchorage independent growth. Okay. In terms of tumor suppressors, which one's a caretaker? You have BRCA1, BRCA2. These are all the other ones, gatekeepers. Okay, chromosomal translocation can cause a cell to become cancerous by putting a proto-oncogene in an active region, creating a fusion protein, knocking out a tumor suppressor, all of these. Which of the following proteins is not made from proto-oncogene? That would be this one, because it's a tumor suppressor. Right? All of these are pro-growth regulators, so their genes are proto-oncogenes. Activation of telomerase is necessary for cell division. False. Cells can divide without it, but only so many times before they end up dying. Right? So they can divide without it, but they will. Their replicative potential is increased. HPV virus. In addition to activating RB, inactivates blank to make the cell cancerous. It's going to inactivate P53. What is directly activated by TPA? I corrected this, so I just said what's activated by TPA, but directly is more specific. It's going to be plasmin. It's turned on by it. TPA, PA means plasminogen activator, so that can't be the answer. MMPs can be activated by plasmin, and focal lesions is just a fourth answer. Which of the following is not a tumor suppressor? AKT, kinase, right? GAP turns off RAS. APC is part of the destruction complex. P10 we talked about above. Burkitt's lymphoma is caused by chromosomal translocation. No, that's the same one. Putting MYC in the region of the heavy genes. That's what it is. And the way I remember that is this is a lymphoma, and this has to do with white blood cells. So we did 10 out of 10. The crowd goes wild. Okay. All right. So let's talk about the questions. I'd like to get a little feedback from the crowd on these, if we can. Oops. It's driving me nuts. Just leave it alone. Whatever. Can't all go away. Okay. See if we can get a little participation here so I don't just give all the answers like I usually do. 3 changes in cells that allow them to invade surrounding tissues can be described as letting go, crawling, and clearing. What are the molecular changes that allow each to happen? So letting go, what does that mean molecularly?
The focal adhesion gets messed up.
And what else?
The anchorage dependent thing also.
Right. So
I don't know exactly how it happens in terms
Yeah,
of like what
that's
happens,
not
but
important, but you're right. Focal adhesions let go, which is one thing. What's the other thing that's keeping cells in the tissue?
could you-
What?
The adhesion molecule, like the integrity.
Adhesion molecules, right? The the cadherins, cell adhesion molecules. Yeah. So both of those are going to be turned off or in some way caused to let go.
Is it a similar mechanism as used by the sister chromatids when they are separated in what it is in the?
I don't think so. No. That's a pretty specific mechanism. Yeah, no, I think it has more to do with like, I don't know it specifically, but I would assume it has something to do with like phosphorylation of the intracellular domains that then changes the conformation of the protein and it lets go on the extracellular domain. I don't know. I would assume it had something to do with that though, because you got to figure it's being regulated by the cell. It has to be in cell crawling so it can grab and let go. So probably has something to do with that. Yeah. Okay, crawling. Who wants to crawl?
I think that involves the reorganization of the cytoskeleton, like RAC, RO, CDC42, and like particularly like the actin cytoskeleton, and then the regulation of actin binding proteins like filament and gelsolin.
Exactly. Yep. Yep. That's Patrick, right?
Yes,
That
sir.
Patrick? Okay. Yep. So we're talking RAC, RO, CDC42, everything we talked about in chapter 3 on cell crawling, that gets activated. And like he said, filament and gelsolin and all those ARPs. Clearing a path, how does that happen?
Breakdown of the extracellular matrix.
Right. And how's that going to happen? Breakdown of extracellular matrix.
I mean, I'm assuming it almost has a little bit to do with letting go as well, because like you're breaking down those focal adhesions and clearing. Yeah, I don't know.
Maybe, but there's a specific mechanism. It was actually
And I,
one of the multiple choice questions. Yeah, go ahead.
isn't it when the tumor cell releases TPA, which activates plasminogen to plasmin, that activates like MMP9, which breaks down the collagen?
Exactly. So the tumor cell is releasing tissue plasminogen activator, which is turning plasminogen into plasmin, and then plasmin activates the matrix metalloproteinases. Nice. Describe one proto-oncogene whose overexpression would result in cancer. What other kind of mutation would have to happen to allow this cell to survive? So first, just name a proto-oncogene whose overexpression would cause cancer. What? MYC. MYC's a good one, right? We make too much MYC, the cell's going to divide. What else are you going to have to do once you increase MYC for that cell to survive and become cancerous?
Stop apoptosis.
Okay, block apoptosis. How might you do that? What's a way to block apoptosis?
Increase BCL2.
Increase BCL2. That's one. So we're overexpressing MYC and BCL2. What else? Let's talk about this because there's tons of answers for this one.
Inhibit P53.
What's that?
Inhibit P53.
Right, just like our HPV, right? Inhibit P53. What else? Think about that. What does P53 do? So basically, we're reviewing what we talked about in the last class on, not the last class, the last test on apoptosis. So think about the apoptosis mechanisms. What would you do to inhibit apoptosis?
Block Cas9.
Yeah.
Block caspase 9.
Yeah.
Okay, yeah. So mutate out caspase 9, so then that internal pathway is activated. What else? Pretty much you can take any protein in those pathways and just say you're going to increase it or decrease it. So Tilak increased, is it Tilak?
Yes.
Okay, increased BCL2, right? Or you can knock out your pro-apoptotic BCL2s. Like that is pretty central to a lot of the mechanisms. BID is central to 2 mechanisms. Knock those out. You could, what else? What
Would
about
AKT
P53?
be-
What's that?
Would AKT be too far upstream to like cause a specific mutation? Because it's like, because I know it inhibits and allows for most of some of the processes. So is it too far up in the pathway to?
No, but the only thing you're right. The only thing I would correct is AKT would be a proto-oncogene. So that'd be the first part of the question. So if you overexpressed AKT or made a constitutively active AKT, that would, and that would also inhibit your apoptosis, right? Because it phosphorylates bad, phosphorylates FOXO1, and all those other pathways that it's promoting. Okay, I guess that's good enough for here. But bottom line, with a question like this, it's so open-ended because there are hundreds of proto-oncogenes. And as long as you know for the second half that you have to block apoptosis too, we've got, at least a dozen proteins there that we can talk about mutating. What might be the consequences of the loss of the ATG1L, which is critical for autophagy? All right, so this question sounds complicated, but really what I'm saying is, what happens if you don't have autophagy, right? I make it a little bit sneaky by throwing this protein in. And you're thinking, what's going to happen with that pathway? If we knock that out, well, it's just not going to happen. So what happens if you don't have autophagy? So again, the question is not what is ATG 16.L, but what is autophagy? Anyone know? Why is autophagy important?
So that the cell doesn't become cancerous. If it's like damaged beyond repair,
Mhm.
then it can do other things.
Not really. We're not talking about cancer with this.
Okay.
We've got 2 reasons to activate autophagy.
Like lack of nutrients.
Lack of nutrients is one, so the cell starts eating itself to survive. What's the other?
Is it just housekeeping?
Housekeeping, right? Housekeeping. So you've got unfunctional or non-functional mitochondria. You got to get rid of it. So those are the 2 things that can activate it. So what would happen if you didn't have autophagy? You'd probably get a buildup of a lot of crap in the cell, big bunches of proteins that are attached to each other for no good reason. And the cell would eventually die because it would become very inefficient, That was a good one. No one knew that one. Okay, why is it dangerous if apoptotic bodies are not efficiently removed? So I talked about this when I did chapter 11.
Won't they get recycled and continue being used?
Normally your macrophages are going to come in and eat them, right? And then recycle them. But what happens if there's more apoptosis than the macrophages can handle, or macrophages are not doing their job and apoptotic bodies start building up?
They'll probably just start attacking healthy cells that don't need to be.
Not really. The specific thing I read in the book, which I wrote, was it's going to cause inflammation. Okay. And inflammation is a bad thing. So if you got a whole bunch of apoptotic bodies building up, then they're going to stimulate inflation. Inflation is bad too. But inflammation, we want to, decrease that. A friend of mine who's a cardiologist said inflammation is probably one of the worst long-term things for our bodies. The whole plaque formation in your arteries is an inflammatory process. So, okay, matching. Oh, I like this. I get a kick out of this pathway, this autophagy pathway, because there's so many different types of cell division, cell signaling going on here. So activation of BCL1 by UNC51-like kinase. Is that PI phosphorylation, ubiquitination, or protein phosphorylation?
I believe it's protein phosphorylation.
I believe you're right. I can't change it. Yeah, protein phosphorylation. So it's a kinase, but remember you got two different types of kinase. One can cause phospholipid phosphorylation. So DSP3 causes binding of WIPI to the isolation membrane. Is that ubiquitination or PI phosphorylation?
PI phosphorylation.
PI phosphorylation, right? VSP34 is a PI 3 phosphate kinase, so it puts a phosphate on the 3 position of the inositol. And then ATG is going to be bound to the membrane by ATG3. And of course, it's ubiquitination because that's what's left. So the thing about this pathway that makes it difficult to remember or to memorize is that ATG, you want to think of ATG as ubiquitin. It's got a very similar structure to ubiquitin, and it's going to be attached to phosphatidylserine, is it, or ethanolamine? And ethanolamine. It gets attached to phosphatidylethanolamine by this, which is again structurally similar to an E3 ubiquitin ligase. And here's another annoyingly named protein. You think they'd give it a new name once they figure out what it does, but this is that PI PI kinase. What is the microscopically evident determinant of whether a tumor has become malignant? So you're looking at the tissue in the microscope. How can you tell if it's malignant or just dysplasia or something like that?
Isn't it if it breaks the basal lamina?
Basal lamina. I had a penny every time I said basal lamina. Okay. What are 2 ways that a chromosomal translocation can lead to cancer? Ooh, what the hell was I thinking here? I don't even know the answer to this one.
The MYC translocation one.
MYC translocation, right? Putting a proto-oncogene into an active region of a different chromosome. What's another thing? We did talk about this today already.
The Philadelphia chromosome one.
Yeah.
The ABL and
Yep.
the other.
And if you wanted to say that, you're right. That's what I'm thinking of. If you wanted to say it in a more generic way, like how does chromosomal translocation cause that cell to become cancerous? You know, you're giving a specific example, which is great, but more generically, what might you say?
Yeah, like the retrovirus entering in proto-oncogene and then mutating it, that causes.
No, we're still talking about chromosomal translocation.
Oh,
I
okay.
guess what I'm trying to say by generic is rather than giving a specific example, then say we can make a new chimeric protein by chromosomal translocation, and that can cause an oncogene to form. Okay, you know what I mean by more generic? Like rather than specific to this one specific gene, this is the kind of thing that can happen and can apply to a lot of different problems.
If there's an abnormal chromosome division, does that happen as well?
I'm sorry, say
If
it.
there's like abnormal chromosomal division, like 23
Division.
versus like,
Yeah,
does that happen?
I'm not sure I know what you mean.
So
Abnormal.
there is a thing called molar pregnancy, which is kind of like cancer, but it's exactly not
Metastasizing?
Yes.
Yeah, I don't know anything about that.
Okay.
Yeah, sorry. You're always finding that thing I don't know. Yeah, you ask good questions too. Okay, I appreciate it. All right, that's it for that quiz. Any questions? So I'm probably only gonna cover one more thing on the next exam. We still have like 4 weeks yet to go. I've got 2 more things I could talk about. One I'm definitely gonna talk about. The other one I'm debating. So the next thing I'm gonna talk about is signaling in early embryo development, using Xenopus as an example. So I absolutely love it. I think it's so cool. It shows you how different hormones can cause different types of differentiation and how that's controlled during development. The other thing I may or may not talk about is internal membranes, the rough ER, the smooth ER, the Golgi, and all of that, how you make proteins. It's important. I don't know how important it is for you guys as medical aspirants. And I'm thinking about doing, instead of covering that, is having the exam a little bit earlier and then doing MCAT review. Would that be something you guys would be interested in? We could review cell biology and physiology for the MCAT, mainly just doing questions in class and working on that. Sound good?
Yeah.
So if we're going to do that, I'll have to move the exam up maybe next Thursday through Sunday, or Wednesday through Sunday. Someone wanted Wednesday last time. That sound reasonable or no? I'll send an email out to everyone and make sure that everyone's cool with it, because I don't want to just move an exam up that far without getting input from the class. But I think it's going to be a better use of our time to do MCAT review than talk about co-translational insertion of proteins into the rough ER. I don't know. I love it, but it's like a blow. Okay, so let's do that. I'll send you guys an email, and hopefully I'll get some feedback and we can decide what we're going to do with that. But as long as we're here, let us gastrulate some embryos. All right, Xenopus laevis. Xenopus is the African claw-toed frog. You can buy them, I think, in the US to have in your aquarium, but if you have one of these in your aquarium, it's the only thing you're going to have in your aquarium soon. These are voracious little guys. I used to do research with them. We would do embryo research with them, and then one time we had some embryos left over, so we just let them grow, and we ended up with 12 tadpoles, and then we had 12 little frogs, and then we had 8 little frogs, and then we had 2 medium-sized frogs, and then we had 1 big frog. They just ate each other. It's hilarious, I guess. I don't tell Pete I said it was funny, but it was kind of. All right, so what are we talking about here? The first thing, the main thing I want to talk about is gastrulation. And what's the most important thing about gastrulation? Louis Wolpert said it was the most important event of your life, which is true. If you didn't gastrulate properly, you wouldn't be here. Gastrulation is going to give us 3 axes. So if you look through the animal kingdom, you can look at really primitive animals which don't have any axis of symmetry, right? They go off in every possible direction. Then you've got radial symmetry type animals where you've got, there's like a central axis and it looks similar all the way around that central axis. That's one axis of symmetry. 3 axes of symmetry then is what vertebrates and chordates have. So we've got back and a front, dorsal ventral, head and a tail, which is anterior posterior, and then left and right into and out of the board. And so there are an awful lot of animals that have this symmetry. Again, chordates, anything with a brain and a spinal cord, or not spinal cord, spinal cord. Not necessarily vertebrae and a spine, but spinal cord anyway. So how does it come about, which is what we're talking about here. So if we look at our Xenopus egg, okay, a Xenopus egg is brown on top and yellow on the bottom. And now this lets you know that these upper cells are going to become this is the animal pole, and this is the vegetal pole. The vegetal pole will become endoderm, and the animal pole will become ectoderm. And the reason for that is we've got maternally deposited proteins and amino acids, or proteins and mRNAs that are attached to the cytoskeleton in these areas, and so they're restricted there. So when this thing becomes fertilized, it's already got two tissues set up already. So your animal pole is fated to become skin and nerve, and your yellow part, it's yellow because it's got a lot of yolk in it. That's where the energy comes from, and it will become endoderm, which is gut, or the whole GI tract, respiratory system, liver, pancreas, thyroid, thymus, bladder, and urethra. So we still don't have anything here to help us move around. So we'll get there eventually. That's what this lecture is about. It's like, how do we get mesoderm and how do we gastrate? So once the sperm enters, the cell becomes a zygote, and now it's active, and it will divide, I guess you would say longitudinally here, to give you 2 cells. Then it divides again to give you 4 cells, and then the third division goes horizontally here, or equatorially, I guess you would say, and that gives you 8 cells. So now at the previous stages over here, you've got both endoderm and ectoderm together in one cell. This is the division that separates ectoderm from endoderm. So at the 8-cell stage now, you've got them separated. And then division continues and continues. The ectodermal cells divide faster than the endodermal cells, but eventually you end up with a ball of cells. That would be a morula. And then when it divides further, it starts to pump fluid into the center of it, and then you get a hollow ball of cells, which is your blastula. Okay, let's watch this little video here. There's your neurula. You can see the spinal cord developing and everything. Okay. That's our gas, that's our whole process there. All right, so let's talk about what's going on inside the embryo as it's going from egg to gastrula. That's what we're most interested in. Okay, so here's our animal pole, and blue is ectoderm. We skipped a step here, which is the production of mesoderm, which is the main thing I want to talk about in this section, because that's where our signal transduction is important. So let's watch this gastrulation just to see how it happens. How many people have had developmental biology before? Anyone? No? A little bit? Okay. So here's your blastocele, which is the hollow part in the center. The marginal zone all the way around the embryo is going to be mesoderm. And then the yellow here is yolk, and that is endoderm. So what's going to happen is that one side of this hollow ball of cells, it looks like it's got radial symmetry, like there's only one axis of symmetry, but actually it's slightly different over here. We'll talk about why later, but these cells start to develop into these bottle cells. And when that happens, these cells are going to come down, hit those bottle cells, and go up. So here's our bottle cells here. We've got the leading edge here of our mesoderm. This is all mesoderm. Hits these bottle cells and it curves and goes up. And when it goes up and in, it's going to take these endodermal cells with it. So what we're doing here is developing 3 layers of cells. We've got ectoderm on the outside, mesoderm here, and then endoderm here. This thing is going to turn, move up, it's going to crawl along the inside of this blastocele. So these cells are coming in, and mesodermal and endodermal cells are going to crawl around that blastocele. And here we are. Cells come in, and you've got 3 layers here, ectoderm, mesoderm, and endoderm coming up. And notice you've got a gap here in the middle of the endodermal cells. And that's the archenteron. So the archenteron is the primitive gut. This will be the primitive anus over here, right? This will be the anus. Eventually, as this goes forward, this will be the anterior end. So this will be the mouth and open at the anterior part of the embryo, and this would be posterior over here. And here we've gone further. The archenteron is bigger. The blastocele is going away. And we've got the yolk plug over here. So you can see now we've got 3 layers of cells, ectoderm, mesoderm, and endoderm. And that's what is giving us our axes here. So again, getting back to the whole idea of making a new axis. Here, we've got a single axis, top versus bottom. And here we've got anterior, posterior, dorsal on top, ventral on bottom, and left and right in and out of the plane of the picture there. So the key thing here again, we've produced 3 axes where we only had 1 before. And at the same time, these things are crawling up. The mesoderm and endoderm are crawling up around this way. On the outside, the ectodermal cells are dividing really, really fast. And they're dividing really fast and sliding down over the whole embryo. So by here you can see pretty much the whole embryo is covered with endoderm, which is going to be skin. So cool, we've got skin on the outside of our body now. That process of the ectoderm dividing and covering everything is called epibole. Okay, epibole. And so it kind of starts at the top and it goes around and it all converges over here. All right, so this is late gastro. And we're not going to get into neurulation, but what happens now is you get interactions here between the mesoderm and the ectoderm that's going to convert some of this ectoderm into neuroectoderm, which will then become the brain and the spinal cord. But we're only going to go as far as gastrulation here and what is causing that. So let's just review a little bit here. So at this late at this gastro stage here, you've got endoderm, you've got your archenteron, which is the primitive gut, endoderm on both sides of that gut. Then you've got mesoderm in the middle and ectoderm surrounding everything else. And so this endoderm then is the gut, respiratory tract, and everything, like I said before. Ectoderm is going to be skin, but also nerves, especially right up here. This will be neuroectoderm. And then mesoderm, I used to teach in the Kaplan course for MCAT review. They called it meansoderm because it's the means of getting around. Everything mesodermal has to do with movement here. So you've got your skeleton and your muscles, which are moving the body, circular system, moving blood, excretory system, and reproductive system. So that's what your mesoderm is going to become. What I want to show here, I guess, what's going to be really important to us in this class is dorsal lip, which is on top of the blastopore, and the ventral lip, which is the Newcoop center and the Speeman organizer. Speeman Mangold organizer, I should say. All right, let's watch this. This woman does a really good job of going through this process. Okay. Does that make sense? Is that good? I'm not going to go much further today, but let's talk a little bit about induction. I'll talk about this again on Wednesday. This is really important. This is exactly what I want to talk about embryo development for, because this is where the cell biology comes into play. So to induce, basically when we say we're inducing something, what's happening is a hormone or hormones are activating new transcription factors to be turned on. Remember, every cell's got the same genes. The type of cell you have depends on what genes are being turned on. So here we're activating new transcription factors. These transcription factors are then going to make new proteins, and the cell type is changed. And also the fate of the cell, like what is it eventually going to become? The idea with cell fate is it's not just what hormone it got hit with, but all the previous hormonal interactions that led it up to this stage before this hormone hit it. I'll talk about that some more on Wednesday, but differentiate then is when you're changing from one cell to another. So you induce the cell to differentiate and becomes different. And typically differentiation involves getting more and more mature, more like a mature cell that's going to do a function in the cell. So, you know, in these early cells with the ectoderm, endoderm, and mesoderm, their job is just to keep dividing and make more cells. And all the energy is coming from the yolk over here. And then eventually they go from, you know, embryonic cells dividing to adult cells that are contributing to the physiology of the organism and keeping it alive. Now, when I say about the changing the fate and stuff, what I mean is I love this picture. It's kind of like you've got balls rolling down this platform here with different pathways that they can go. Every time they hit a nexus point, they go right or left. So in this case, it's going to go right. When it hits this one, it goes left, and it ends up there. But it could have gone this way and here and go that way. So the point being, each of these nexus points is when a hormonal influence happens. And so like an ectodermal cell will get hit with a hormone and become a mesodermal cell. That mesodermal cell will then get hit with a different hormone and will become an anterior mesodermal cell. And it'll keep going until it becomes, you know, a mouth part or something, you know, a part of the anterior mesoderm. So each step then involves hormones activating different proteins, turning different proteins on, and how it's going to respond to any particular hormone depends on what hormones it's already seen. How did it get to where it is, and how's it going to respond to that? So this is what we're going to talk about starting Wednesday. I think I want to stop here for today. Any questions about that? Go back to the big board. Patrick is here. Right, I'm not going to do that. Okay, no more questions, comments? We all good? Okay, don't forget to get your exam 3 exam corrections in. I'll take care of that hopefully today or tomorrow. What else do I want to say? I'll communicate with you via email again to talk about what we're going to do going forward. Any questions, Brittany?
Yes. Hi, Dr. Slish. Yeah, could you explain one of the quiz questions about Burkitt lymphoma? I kept going back and forth between the Epstein-Barr virus, thinking about like my clinical training and how we were always taught, you know, always take into consideration human herpes virus for Epstein-Barr as causing Burkitt's lymphoma versus like the MIC. So could you review that question for me, please?
I can, but frankly, it sounds like you may know more than me about this. I'm just going by what was in the textbook there, so I could be wrong. Burkitt's lymphoma.
I mean, the MIC part is right, but then I was just wondering why, because it seemed like both of those would have been.
Oh, so you think Epstein-Barr could be also an answer for Burkitt's lymphoma?
Yes.
Don't know. We live in the internet age. We don't have to wonder about anything.
Yeah, because I remember when in clinicals, like they taught us about HHV 1 through 8, and any of them that had like any malignancies associated with them. Like when we think about HHV 8, we think about Kaposi sarcoma, but HHV 4, we think about Burkitt's lymphoma.
HPV is linked to the pathogenesis of lymphoma such as Burkitt's. And it does say Burkitt's there. I think I could be wrong. There may be two different type of Burkitt's lymphoma, because I know there's one that is caused by the Epstein-Barr.
Right, the endemic form.
Right. Let me bars. Yeah, there it is, Epstein-Barr virus infection of it. So what is with the, all right, let me look into that, because that, what I said, let's bring that up. I make up a lot of things, but this isn't something I made up. Just kidding. Burkitt, where's Burkitt? Here. Yeah, let's look into this. This is from the textbook World of the Cell by Pearson.
And I'm not
Yeah.
saying that's wrong. I just thought it was two right answers, but I wasn't sure what to prioritize because we weren't, like it just said Burkitt's lymphoma is caused by.
Yeah. Yeah, and Epstein and Barr worked with Burkitt in Africa on that project. So yeah, let me look into this. Maybe this, I don't know, it's in the textbook. Usually textbooks are not the cutting edge. So let me look into that. You're probably right. So I should probably give everyone credit for that who did either one, right?
That's
This
what I
may
was
be true.
thinking. I just didn't know what, I mean, there wasn't any kind of other context clues to prioritize, you know, which
The other.
answer choice, but the, yeah.
Hide
So I
myself
was just.
black context clues. All right, so let's look into that after class. Any other questions or comments?
Oh,
All right, so
Dr.
yeah, go
Slish.
ahead.
So you said that if you, and I know you're going to send an email to us, but I kind of missed part of it. You said that if you moved up the exam, you would do like MCAT high yield review.
Yeah,
Okay.
yeah.
Basically
So, 'cause
from,
we still have 4 weeks left. If we don't cover new material, we'll be done in 2 weeks. That'll give us, 4 more classes to do something else useful. And I think that would be a good thing to do.
okay. But if you did cover new material, would you add a 5th exam or what were you thinking?
No, I would put it all on the 4th exam. I don't want to have
Okay.
another exam. I think you guys
Gotcha.
have enough. I think too, you could tell me if I'm wrong, but if we have an exam next week, second half of next week, is it going to conflict with anything else you have? Any other exams going on now? That might make it a little bit nicer for you guys too, instead of
Yeah.
having them all right at the end of the semester. Sound good?
I'd let my other classmates chime in there because I think some of them have a heavier class load than I do.
Okay, I will, you can set up polls on Brightspace. I'll do that. I'll give everyone the lowdown and then set up a poll so you can all weigh in on that. Okay?
Okay, thanks, Dr. Slish.
Sure. All right, we ready to call it a day? Go home? Get a nap? Or is that just me? All right. Later. All right.